Today's topic is genetic engineering for improving quality and productivity of crops. Genetic engineering is the process of using modern biological techniques to modify genomic DNA at the molecular level, thereby changing the genetic characteristics of recipient cells to achieve specific effects. In 1983, genetic engineering was first applied in plants and herbicide-resistant tobacco was produced, starting the era of plant genetic engineering. The importance of optimal nutrition for human health and development is well recognized. Adverse environmental conditions such as drought, flooding, extreme heat and so on affect crop yields more than pests and diseases. Genetic engineering technology, transgenic technology and gene editing technology. 1. Transgenic technology. Transgenic technology is a method that transmits a piece of DNA in line with the wishes of researchers to the target cell through physical, chemical, biological, or comprehensive methods and integrates it into the target cell genome to achieve its expression. The most commonly used transgenic techniques in plants are Agrobacterium tumefaciens transformation and biolistic technology. Gene editing technology. There are many types of gene editing tools, such as zinc finger nucleases, FFN, transcription activator ligator, effector nucleases, and clustered regulatory interspaced short palindromic repeat CRISPR-associated 9-protein twisting. ZFNs are the fusion of artificially modified zinc finger protein shifts DEF, having specific binding activity, and FOC endonuclease, having non-specific cutting activity DUNDE. Several amino acid residues on the A helix of ZFP play a direct role in the recognition of the target site and can pair with bases adjacent to it. For example, the E136 locus on the A helix of ZIF268, a kind of ZIF, can directly recognize and bind to 3 adjacent bases on the target sequence. In addition, the binding domain and cutting domain of the FOC endonuclease can be separated, and the non-specific cut-and function of the FOC endonuclease can be obtained when the binding domain is removed. Finally, under the action of FOC dimer, the target site will be cut and DUTs will be generated. ZFNs, as the first generation of gene editing technology, have been successfully applied in animals at first and have attracted wide attention. Subsequently, ZFNs were applied in corn, rick, and other crops. However, there are inherent flaws in this system. For example, the inter-ZFP context effect, that is, it is difficult to achieve high efficiency by simply linking specific ZF together. In addition, there are shortcomings such as unsuitability for multiple editing, high off-target efficiency, and high cytotoxicity, which are gradually being overcome by the later gene editing tools. Therefore, its application in crops is not well developed. 2 talons. The structural difference between talons and ZFPs is that talons use the transcription activator like effector tail as their binding domain, and the binding function mainly depends on highly variable amino acids at the 12th and 13th position on the tail. In combination with them, a single base can be specifically identified. The deciphered recognition method can meet the needs of arbitrary base In this system, the non-specific cutting domain is still FOC endonuclease, and a pair of talons will form a FOC endonuclease dimer after binding to the target site. Talons are more targeted, less cytotoxic, have no contextual effect, such as the one in ZFPs, and can be assembled in a modular manner. Currently, they have been successfully applied in rice, corn, wheat, and other crops. However, talons' binding sites are restricted by guanine nucleotides and are not suitable for multiple editing or highly methylated sites. But highly methylated loci are very important in gene regulation research, and multiple editing is of great significance in studying the interaction between genes and improving editing efficiency, so the application of TALENs, Cas9 system, the widespread application of gene editing technology in crop improvement at present largely benefits from the emergence of the Cas9 system, which is derived from the immune system of bacteria and archaea. The Cas9 system was synthesized by connecting single-guide RNA and Cas9. The cRNA can bind to the target site after specific modification. The RUFC domain of Cas9 protein can cleave the DNA strand with protospacer-associated motif E sites, and the DNA strand complementary to cGNA is cleaved by the HUNH domain of Cas9 protein. The cutting site is near the PAM site. Follow for more informatic videos.